And we're live. Hi, everyone. Thank you for watching this Science on Google Plus Hangout on Air. We're doing this in association with Mosaic Science, and we have this Mosaic Science, if you don't know, is the Wellcome Trust magazine, and there's a whole bunch of amazing authors who write really in-depth pieces for it. And my guest today is Sue Armstrong. Hi, Sue.
Hi.
And Sue, can you tell us a bit about yourself? Because we'll be having a Q&A about what you wrote about.
Okay, basically I'm a science journalist. I started out as a science journalist, writing and broadcasting, and always a freelance, and particularly working in foreign countries. I did a lot of work on AIDS and so on. And a few years back, I got involved with writing about P53. And I also decided that I really wanted to spend more time focusing on something rather than doing little short stories for newspapers and so on, science stuff. And so I started writing books. And my latest one is on P53, the gene that cracked the cancer code. And then the Mosaic, I got in touch with Mosaic, and we decided to come up with an article on a new angle of the P53 that hasn't been covered so much in my book.
That was a really, really cool book. And yes, I really loved the article you wrote as well. So can you tell us a bit about what is P53? What does it do in humans and so on?
Okay, P53, basically it's a tumor suppressor gene, which means that its main task is to see that when our cells divide as part of the regular maintenance and growth in our bodies, that they do so without making mishaps, without making mistakes. And very basically, this means seeing that the DNA is not corrupted in the normal hurly-burly of living, and when cells are dividing, it's copied faithfully from the mother cell to the daughter cell. Because if the DNA is scrambled, this can trigger cancer at any time. So P53 will notice this while the cell's dividing, and if it sees that the DNA is scrambled, it'll stop the cell mid-cycle, and it'll send in the repair team to mend the DNA. And if the DNA is beyond repair, it instructs the cell to commit suicide, and so clears out the thing. And it's an extremely important gene, because they reckon that in most human cancers, P53 itself is not functioning, either because it's mutated or because some other mechanism is stopping it working. So the key protection against cancer is not working in the majority of human cancers. And just one other thing, you realize how terribly important this tumor suppressor is, because there are a number of others as well. They reckon that in the normal span of a human lifespan, our bodies undergo something like 10,000 trillion cell divisions. And it takes just one cell which has become scrambled and which has escaped the sort of surveillance mechanism. That can trigger cancer. It can start a tumor forming. And it's P53's job to see that and scupper it before it can get going. So it's a mighty important gene.
Yeah, because cancer really is a disease that arises with one single cell. And what you're saying is P53 is what makes sure that one single cell doesn't become cancerous in the first place.
Absolutely. Yeah. That's its main task.
Yeah. So your article for Mosaic was titled Brazil's Cancer Curse. What does Brazil have to do with P53?
Well, there was a young doctor in Brazil who noticed that she was a cancer geneticist, and some of her first patients who came had what she thought was what she identified as Li-Fraumeni syndrome. Now, Li-Fraumeni syndrome is a condition in which somebody is born with mutant P53 in every cell of their bodies, and it means that they're incredibly prone to cancer. They're very vulnerable to cancer. And in the classic Li-Fraumeni, somebody is almost 100% likely to get cancer at some point. And it was considered to be incredibly rare, this condition, because of course, if people get cancer very early, they don't tend to survive up to a time when they can have children and pass on the gene. And so it tends to die out naturally. And it was considered terribly, terribly rare. There were very few cases in the medical literature. But this young doctor in Brazil, this young medical geneticist, she was at the beginning of her career, she kept seeing these patients coming in, and she thought, looks like Li-Fraumeni. And she thought, this is crazy. There can't be so many like this.
Yeah.
And some of her colleagues said to her, you should go to one of the cancer conferences and just take some of your cases there. And she took some of her case notes to a conference in France, where she met a guy called Pierre Hainaut, who is, he was then working at the WHO, World Health Organization's Cancer Center in Lyon. And he was the curator, the custodian of a P53 database, which made a note of all the P53 mutations that were mentioned in the medical literature and the tumor types they were associated with. And he knew about leishmaniasis, and he knew it was terribly rare, and he was fascinated by her studies. And so he said to her, would she go back and collect some of the blood from her patients and bring them back to Lyon, and they could test to see if they did have a mutation in P53 and what the mutation was. And sure enough, they discovered that they had. And when they did further studies to see how far this gene had spread, they reckoned that there are hundreds of thousands of people in southern Brazil who are all carrying an identical mutant P53. But it's slightly, it's an uncommon mutant. It's not the classic, most of them don't have the classic leishmaniasis mutant, which means that they tend to get cancer a little bit later than the classic leishmaniasis, and that it's not 100% certain they're going to get it. It's slightly less. It's sort of 70%, between 50 and 70%, which means that a lot of people do get through the reproductive cycle, and so it can be passed on. And it has just spread massively throughout southern Brazil. And so there are a lot of these families with a cancer predisposition syndrome because they've got a particular mutation in their P53.
Yeah, I can absolutely imagine how some of these families can feel like they are cursed, because, you know, you could have a mother that passed away really young with ovarian cancer, and then, you know, a child gets another form of cancer. What's going on? How can I draw such bad luck so often? Do you think, so these families find out then that there's this mutation and there's a reason for their bad luck. Do you think that gives them more closure? How do they react to that?
Well, I mean, that's actually a very interesting question, because, of course, you know, everybody's different. You've got some people who were really deeply curious. What on earth is this? They didn't believe in the curse idea. They thought there must be something behind it. And so they were mighty relieved to find that there was a cause for it, a physical cause.
Yeah.
And one particular family of this doctor in Brazil, Maria Isabel Achatz, one of her families was a huge family, and there was a lot of cancer in them. And they all came together in somebody's kitchen, and Maria and Pierre were both there talking to them and explaining what they knew about it and the blood results and so on. And some of them were really curious, and some of them were really freaked, because if their blood had proved positive, then some of them felt, well, you know, I'm doomed. Others realized that this was actually a lifeline, because what it meant was they could have screening sort of every year for a sort of regular screening program to catch anything right at the very beginning, and that it was actually a way of keeping them alive. And so some of them were excited by it. There were others who would take the blood test and then be too scared to come back for their results. And I met two cousins. One of them was really, really freaked by it, and she said she'd watched her mother die of cancer over several years. And every time she had to go to the hospital to get her screening, she felt really, really unhappy about it. And it dogged her all through the year that she'd got to go for the screening. And her other cousin thought, yes, jolly good, I can go, and then I get a clean bill of health, or they find something tiny and they can get rid of it. So people have completely different ways of looking at it.
Yeah, that's really interesting. And I guess it's one of those questions that you never really know until you're in that situation. You don't know how you're going to react. Yeah. So P53 is a gene that we've known about for a while. It's not new. Why are people still researching it? What's still left to uncover about it?
Well, I mean, that's the fascinating thing about P53. It was actually discovered in 1979 by David Lane in Britain and three other people, Arnie Levine in the States, and four people discovered it at exactly the same time, and all completely unknown to each other.
Yeah, and it was
and
called the molecule of the year, right?
It was the molecule of the year, that's right, yes. Because they thought this was really exciting. Yeah. But then for a long time, when they started to look at it, they thought that it was a gene that drove cancer, that caused cancer. And they'd already found a few of these and they weren't that excited by it, so people began to drift off. And then they discovered it just didn't play by any of the rules, so they went on being thoroughly curious about it. And suddenly they realized that in fact it was a tumor suppressor, and tumor suppressors had been hypothesized. Nobody knew if they really existed. There was just a suspicion that they did. Somebody suggested they might do, and this was the second one that was found, and so people got very excited by it. And since then, they've discovered that it's unique among tumor suppressors in that most tumor suppressors, if they become mutated or corrupted, that's it. They're messed up and they don't function. P53 has a double life. If it becomes mutated, it can cause cancer. It can actually be a cancer driver. It's got all kinds of different actions at different levels of mutation and so on, different types of mutation. So it's a very subtle gene. And people just are discovering the whole time something new, something new, something new. And they'll look at some other aspect of cancer and suddenly think, my goodness, P53 is playing a role in this as well. And that's where my article in Mosaic came in, that for a long time people had known that the metabolism had a very strong role in cancer, but they knew roughly how, but then they realized that P53 was also involved in metabolism, and it looked as though it was part of this mechanism, but nobody quite knew how, and it didn't fit with its tumor suppressing activities. So it was, it was one of those things on the sort of sideline, and then they began to have breakthroughs and find out roughly what it was doing. And so the whole time you're going into a new area of cancer, and suddenly you find, P53 is lurking somewhere in the picture.
Yeah.
And so you never stop learning about it. It's one of the most intriguing genes and proteins around, it seems to me.
Yeah, and I think the key point that people need to remember is that P53 doesn't do its job in isolation, and it's the fact that it's linked to so many other processes in our body. And cancer doesn't just happen in one way. There's so many different ways that cancer happens. It could be cells that divide too fast, cells that don't know when to die, different things like that. And I think, like what you said, is it's P53 is involved in each of these processes in many different ways. So it's almost like it's like a chameleon. It shape shifts according to what function it does.
Absolutely. But I think you've hit on a really important thing, because a lot of people say, well, if P53 is the thing that stops cancer, and when it's and in most cancers it's messed up, surely all you need to do is screen and see somebody's tumor and see if P53 is messed up, then you know what's happened. But exactly as you say, P53 is at the very center of a web. It doesn't do all these tasks itself. It's a sort of orchestrator of everything, the master switch. It sends in the repair team. It sends in the things which will make cells commit suicide. So it's a sort of master switch of all of these things. And you can have a mess up in the things that send signals to P53, or in the things that P53 switches on, downstream or upstream. You can have a mutation or a corruption, and that will stop the system working as well. So you can have cancers which look as though P53 itself is still intact, and you think, well, what's happened here? But it'll be one of those things. The things which send its signal, or the things that it signals, can be messed up. So it's massively complex.
Yeah, and it's also not guaranteed that if you have inherited a mutant copy of P53 that you're guaranteed to get cancer. It just means you have a higher likelihood of getting it. And likewise, if you have cancer, there's no guarantee that it's because you have a mutant P53. So the two are not always, they don't always go hand in hand.
I think that's right, and I think that's what made it such a deeply fascinating story to tell. That it wasn't a sort of linear progression. Oh, we found this gene. We thought it was this, but now we found it's this, and this is how it works.
Yeah.
Every time they make a discovery, you find somewhere that it doesn't play by the rules. There's something else that they need to know. So it's like a jigsaw puzzle, and you get little pieces. And what I found fascinating writing the book was that there were lots of bits of the jigsaw puzzle, just like when you have a huge jigsaw puzzle spread out on the floor, and you find a little piece, and it's got a piece of the sky, and you've got lots of pieces of sky, you don't know where it fits, and so that piece just gets left. And then suddenly you've got a hole, and you realize that's where it fits in. And that's exactly the story of P53. There's been lots of other sidelines in cancer research that have been done, and people have said, oh yes, fascinating, but so what, sort of thing. And then they suddenly think, hey, this is that little missing piece of sky that we were looking for, and it all comes together. And what I love about it was how it's foxed people. There have been some really great minds involved in this, and they've got it wrong for a long time, and then suddenly they realize, my God, we were going down the wrong path. And it was such an exciting quest, and the people I spoke to were so ready to talk about the blind alleys they'd been down, to explain the thrill of the real science rather than just the thrill of getting there and making big discoveries.
Yeah. So I guess that's my next question to you. What makes a good story? Because yes, it's really cool science, but how do you make it into a story? What's your process? How do you turn that information into a story in such a compelling way, the way you do?
Well, I think from the start, I should actually say that I'm not myself a trained scientist. I grew up in a family full of medics, and I always wanted to be a medic, but I wanted to be a writer, and that was the most pressing thing. And so I went off into journalism, but then of course I drifted towards medicine and science and so on. So when I go to interview people, I say, I'm not a medic, I'm your direct, I'm not a scientist, I am the audience. I'm a very curious person who's prepared to work hard, and I do work incredibly hard so that I go in and find out what's happening. So what I think makes a good story, to me, it's human faces, intellectual mysteries and conundrums that people are teasing out, and relevance, hopefully relevance to the reader. And of course, you know, cancer is there's almost no family that isn't touched by cancer in some way, either friends or family. And so many people are curious about it, and so many don't actually know what's happening in it. And so I start off from that question, and you go and meet people, and you meet such interesting characters, and you know, either the people who are affected by the problem or the people who are researching it. And I think what you have to do is bring out the human faces and the human stories, make it relevant to your readers, and tease their curiosity. That's what I think makes a good story. It's been those have been my guiding principles whenever I've been writing a story. If it excites me, I think, okay, you've got to enthuse your audience as well.
Exactly. And it's so clear talking to you that you're so passionate and fired up about it. And I think that's what makes it such a compelling story, because you're able to almost infect people with your enthusiasm for it.
Well, that's what I hope to do. I mean, you know, I just think it's been such a privilege. I get such a thrill, and I go to interview people, and I come away thinking, wow, why didn't I study science myself? And then I think, you've got the best of all worlds. You know, you can go in there and you can ask people how they do it and what were the good moments and what were the bad moments. I get to live vicariously the thrills of science, but I don't have to sit there, you know, teasing apart little microscopic worms and counting cells and counting genes and so on. So
and also you don't have to deal with the heartbreak of failure, because having worked in academia for a long time, I can tell you there were so many nights where I'd be in the lab at 2 in the morning wondering, what am I doing with my life, because this experiment is just not working. So at least you don't have that.
Oh, I can imagine. I mean, some of the people I interviewed had exactly that, you know, that they've been working so hard. Or one guy had written a paper, and he was so excited about it, and he'd sent it off. I can't remember which journal, but a big journal. And he went off to a conference, and he heard that 2 other people had found the same thing, and they'd got their articles into a really good journal. And he got back and found his had been rejected. And the reviewers had said, you know, this guy is barking up the wrong tree. He may think he's onto a good thing, but we all know P53 is such and such. And he was absolutely heartbroken. His paper had been rejected, but because the reviewers didn't like, you know, they said, no, he's wrong. And, you know, and when he actually told me this story, it was so dramatic. And so, you know, those are the sort of things that I think make a really good story. It's not just cool scientists just finding out things and telling the rest of us.
And I think that's one of the things that I think science communicators should be talking about. It's not just about the successes, but the failures that lie in the way, because otherwise we portray an unrealistic view of what it's like to do science.
Absolutely. I mean, I read something, and I'm going to get the quote wrong, but I thought, yes, that's it. Somebody said recently that scientists, they uncover more that we don't know than that we do know. And
Yeah.
I thought, yes, they you know, it's more, oh yes, now I know what I don't know, rather than, oh yes, we've discovered something new. And I
Yeah.
think that's the truth, isn't it? You
Yeah.
keep uncovering it, and you keep being aware.
Sorry, I think our connection just halted. I think we've lost Sue, but give me a second and I will try to get her back. Please bear with us because we're having so many technical issues with this Hangout.
Yes. That's me again. Can you see me?
Yes, okay. Hi. Hi, everyone. So sorry about these technical issues. I have no idea what's going on with Google, but yes, blame the Google gods. Maybe they're Scottish rugby fans.
Oh. I think it could be, yes.
So anyway, back to P53, where we were. So another question I wanted to ask you is, a lot of times with cancer, it's very easy to switch off something that's switched on. So you can have drugs that inhibit it and things like that. But P53 is something that should be switched on, but it's switched off. How do you reactivate something that's not working? It's not as if you're trying to stop an overactive protein, you're trying to reactivate a protein that's not working. What can we develop in terms of therapies to actually help people with this?
Well, there are, I mean, there's all kinds of things that they are trying at the moment, because several things, either to, if people have got a mutant P53, they're trying to find ways of introducing a normal copy, a good copy of P53 back into people's genes or into people's cells. So there's that line of approach. There's another one where mutant P53 is that the protein folds in the wrong way, and how a protein folds is critical to how it acts in the cell. And if it's folded wrong, it can just mess up everything. And they're finding some very interesting little drugs that are able to refold P53 into the right way. But I mean, these are all, it takes a hell of a long time from finding things that work in lab dishes and all this sort of thing, and even from doing the first clinical trials to it actually being available in the clinic. And those are two approaches that they're using. Yes, and I mean, just several things, trying to actually reactivate P53 that's wrong, or to reintroduce a copy that is normal into cells and so on. But there's a whole lot of different approaches to this, but it takes a long time between finding something that works and actually getting it to the clinic. So there's a lot in the pipeline.
Yeah. My next question is about P53 itself. So we know that it's linked to cancer in humans, but we also know that it's found in other animals. How is it linked to cancer in other animals? Because there was the whole elephant article that was in the news recently.
Well, this is fascinating. P53, because it's such an incredibly important gene and protein in bodies, seems to be in so many things. It's concerned from very, very early on in the tree of human of life. And so it's in lots and lots of species. But one of the great conundrums, and I think it's known as Pito's Paradox, is that if cancer is linked to the number of cells you've got that are constantly dividing, and as we grow and maintain our bodies, and little problems are getting into the DNA, there's lots of opportunity for things to get scrambled or corrupted. That's one of the reasons cancer is more prevalent as we get older, because we're introducing more corruption into our DNA. But then you look at something like elephants, which have got, and whales, and they've got many more cells than us, and yet they don't seem to get cancer as often as us. And in fact, elephants, they say that, studies have shown that only about 5% of elephant mortality is related to cancer, whereas it's up to 25% in humans. So what's going on? And somebody asked, well, a lot of people have asked this question, but fairly recently, people looked at the P53 and discovered that elephants have something like, we have 2 copies of P53 in every cell in our bodies. Elephants have 20. So they've got incredibly strong P53. And the researchers were wondering, well, what does this do? They thought it would be very much better at repairing damaged DNA. And so they irradiated some cells in a dish, elephant cells and human cells, to mess up the DNA. And they expected to see that the elephants were very good at repairing it. And they didn't find that. But what they found was that the elephants almost, their first reaction, instead of just stopping the cells, repairing the DNA and then getting on with cell division, they wiped the cells out. They caused them to commit suicide, just like that. So they reckon that the elephant's strategy for avoiding cancer is to just eliminate cells which look a bit ropey right from the start. So you've got that, and you've got other species which have, there's another one which is very interesting, and you and I were talking about this before. The blind mole rat, which is a very interesting little critter that seems terribly resistant to cancer. And this is a little creature that lives underground in very, not a lot of oxygen, it's blind and all of that sort of thing. And they found in that it's got a mutant version of P53, or a P53 that isn't the same as ours, and it seems unable to cause apoptosis, to cause cell suicide. It does send in the repair teams, and it's quite good at repairing its DNA if something goes wrong, but it's not good at eliminating cells. And so you wonder, well, how does it avoid cancer? But what it seems to have is a mechanism, a sort of compensating mechanism, which the immune system, any cells that do become cancerous that aren't mended properly by the DNA repair things, seem to attract the immune system, and it pounces on them like a ton of bricks and wipes them away that way. So it causes them to die in another form and gets rid of them.
Yeah, it's almost like the cleanup crew that, it compensates for what the fact that the cells don't kill themselves when they should.
But the interesting thing is, this is where the story gets sort of linked to this thing I did in Mosaic, that one reason why it's good for the mole rat to have a sort of weakened version of P53 is because it lives in conditions which are very low in oxygen. And normally this is the sort of thing which would put cells under the kind of stress which would trigger P53. And they don't want, that would be disastrous if it was being triggered the whole time just because there wasn't enough oxygen around for their metabolism. And so it's better to have a weaker P53 and to try and get rid of cancerous cells by another mechanism. So, they're teasing out these little wrinkles the whole time in P53, which is what makes it such a fascinating story.
Yeah. I'm afraid we're approaching close to the time we should stop, even though we had that unintended interruption. Thank you, Google. Was there anything else that you wanted to add? Anything that we have? Well, I think we could talk about P53 all day long, but is there anything that you think we should have talked about?
No, I don't think so. Well, except that I think just one other thing. You were talking about the kind of treatments that we might have that are, real relevance to people's everyday lives. I think one of the great things about P53 is that it has also indicated to people what it's given an idea. It's given an idea of the kind of things that cause cancer. Like it finally nailed tobacco as the culprit in lung cancers, and it's nailed moldy peanuts, or the mold round peanuts that haven't been stored properly, in liver cancer in West Africa, and various things like that. So it's actually it really has clinical relevance already, and there's lots more will come out through the pipeline, and just look for new stories of P53 all the time.
Yeah.
And meanwhile, read my book and go and read the article in Mosaic.
Yes, definitely. I think that P53, it's the gene that keeps on giving. Like, we'll keep hearing really exciting stories going forward as well. So yes, thank you so much for talking to me about this. It's been.
Well, I'm sorry we got the gremlins in our broadcast.
That's okay. I think despite the gremlins, it's such an awesome conversation, and hopefully people who couldn't watch this live are able to watch this recorded. So I'm going to stop the broadcast now, but please stay on for a second afterwards.
Thank you very much.
Yeah, thank you for watching.
Okay,
Goodbye.
Thanks. Bye.